We will continue skeletal muscle fibers. As we said before, that the skeletal muscle fiber has a cell membrane called the sarcolemma, which sends a transverse tubules, and the cytoplasm contains myofibrils formed of myofilaments and contains smooth endoplasmic reticulum called sarcoplasmic reticulum, and also the cytoplasm contains many mitochondria, Golgi apparatus, ribosomes, and the inclusions include myoglobin and glycogen. As we can see, the transverse tubules, this is an invagination of the cell membrane, transverse tubular invagination of the cell membrane into the muscle fiber to surround the myofibril. It conducts the nerve impulse from the surface of the cell membrane into the interior of the muscle fiber. The sarcoplasmic reticulum, which is smooth endoplasmic reticulum, formed of branching and anastomosing network, and on each side of this network there is a dilated tubule called terminal cisterna. This terminal cisterna, formed of smooth endoplasmic reticulum, present on either side of the transverse T-tubule. And this triad formed of transverse T-tubule and two terminal cisterna on either side is present at the AI junction, at the junction between the A band and I band. This sarcoplasmic reticulum stores the calcium for regulation of muscle contraction. It releases the calcium during muscle contraction and sequesters it during relaxation. Again, we can see the T-tubules, which is invagination of the cell membrane to surround the myofibril, present at the junction between the A band and the I band, and on either side of the T-tubule there is a terminal cisterna, dilated cisterna of the sarcoplasmic reticulum called triad. So we have two triad in each sarcomere. The importance of T-tubules for fast contraction of the nerve impulse into the interior of the muscle fiber to stimulate the sarcoplasmic reticulum and the terminal cisterna for release of calcium. When the calcium is released from the sarcoplasmic reticulum, it binds to troponin of actin filaments, leading to the interaction between actin and myosin, finally leads to muscle contraction. So during contraction there is shortening of the sarcomere, and the I band is reduced in length while the A band remains the same length. Also the H band or zone disappears during contraction, as we can see. By electron microscope we can see between the myofibril, this myofibril, and this is another myofibril. Between them there is many mitochondria. This mitochondria and this mitochondria. There is many mitochondria, and also there is ribosomes. This electron dense dots represent the ribosomes. The inclusions present in muscle fiber are glycogen and myoglobin. Myoglobin, which is an oxygen binding protein similar to the hemoglobin. It is red to brown in color, and according to its concentration in muscle fibers, the muscle fibers divides into red fiber, white fiber, and intermediate fibers. This is a comparison between the three types of muscle fiber. As we can see, in white muscle fibers the amount of myoglobin and mitochondria are low, but in red muscle fibers the amount of myoglobin and mitochondria are high, so the color of the muscle fiber here is red due to high amount of myoglobin and mitochondria. The glycogen is high in the white muscle fiber in contrast to the red muscle fiber. The contraction of the white muscle fiber is rapid and of short duration, and the muscle can easily fatigued, like the muscle of the digit and the muscle of the eye, the extraocular muscle. But in red muscle fiber, the contraction of it is slow and of long duration, and the muscle cannot easily fatigued. It is fatigue resistance. It resists the fatigue, like muscles of the back and the muscles of the lower limb, so we can stand for a long time, and the runners can remain running for a long time. Growth of skeletal muscle in length by the activity of satellite cells, which add new skeletal muscles, but the growth of skeletal muscle in width by the synthesis of new myofilaments from ribosomes. Regeneration of skeletal muscle in children by the division of skeletal muscle, but in adult, a small defect can be repaired by the satellite cells. Large defect cannot repair, and it heals by fibrosis. Duchenne muscular dystrophy. It is a disease affects the muscle. In Duchenne muscular dystrophy, there is progressive muscle weakness due to defect in dystrophin protein. This protein binds the skeletal muscle fiber actin filament to its external lamina. The disappearance of this dystrophin and absence of this dystrophin leads to necrosis of the muscle fibers and finally atrophy. Myasthenia gravis. It is an autoimmune disease caused by the formation of antibodies to the acetylcholine receptors on the sarcolemma of the skeletal muscles, leading to inability of muscle contraction due to inability of depolarization of the skeletal muscle. So there is muscle weakness. The manifestation include muscle weakness, inability to lift the arm, failure to maintain an upright posture due to inability to muscle contraction.